Good day everyone, we are Group 4 and we have decided to create a nettable model of the animal cell. We are here to discuss the parts and functions of the animal cell verbosely. I hope everyone is ready to listen as we begin with what is animal cell all about.
What is animal cell all about? Animal cells are eukaryotic cells with a plasma membrane-bound nucleus and organelles. Animal cells lack a wall, unlike eukaryotic plants and fungus. The single-cell creatures that gave origin to the kingdom Animalia lost this trait long ago. Most animal and plant cells are between 1 and 100 micrometers in size, making them visible only under a microscope.
For the first part, we use tomatoes to serve as our mitochondria. Mitochondria are membrane-bound cell organelles that provide most of the chemical energy required to fuel the cell's biochemical reactions. Adenosine triphosphate is a tiny molecule that stores the chemical energy created by mitochondria. Mitochondria have small chromosomes of their own. The second part is lysosomes. We use mushrooms to serve as our lysosome. Lysosomes are membrane-enclosed organelles containing various enzymes that may degrade a wide range of biological polymers, including proteins, nucleic acids, carbohydrates, and lipids. Lysosomes serve as the cell's digestive system, degrading material taken in from outside the cell and digesting obsolete components within the cell.
The third part is peroxisome. We use hot milk for our peroxisome. Peroxisomes are tiny membrane-enclosed organelles that contain enzymes involved in various metabolic activities, including energy metabolism. Although peroxisomes resemble lysosomes, morphologically, they are built from proteins generated on pre-ribosomes and subsequently imported into peroxisomes as complete polypeptide chains, similar to mitochondria and chloroplasts. The fourth part is microtubule. We use beans to serve as our microtubule. A microtubule is a tubular structure of indefinite length consisting of tubulins. Globular proteins present only in eukaryotic cells. Microtubules provide a variety of purposes.
The fifth part is the Golgi Apparatus. We used onions to serve as our Golgi Apparatus. The Golgi Apparatus is a membrane-bound intracellular organelle that plays a crucial role in the trafficking, processing, and sorting of newly generated membrane and secretory proteins and lipids. Golgi membranes have a unique layered structure that allows them to accomplish these functions optimally. The sixth part is the smooth endoplasmic reticulum. We used ham to serve as our smooth endoplasmic reticulum. The smooth endoplasmic reticulum, or SER for short, is a meshwork of tiny disc-like tubular membrane vesicles in the cytoplasm of eukaryotic cells involved in the synthesis and storage of lipids such as cholesterol and phospholipids that are essential in the formation of a new cellular membrane. The lack of ribosomes distinguishes the smooth endoplasmic reticulum from the rough endoplasmic reticulum, or RER, the second leading form of the endoplasmic reticulum.
The seventh part is ribosome. We use chopped carrots to serve as our ribosomes. The ribosome is a micromachine that produces proteins. Unique proteins and nucleic acids make up ribosomes. Ribosomes get their information from the nucleus of the cell and their building components from the cytoplasm. The information encoded in messenger ribonucleic acid is translated by ribosomes. They assemble polypeptides from certain amino acids and transport them to the cytoplasm. The eighth part is raw endoplasmic reticulum. We use ham to serve as our raw endoplasmic reticulum. The raw endoplasmic reticulum is a group of latent sacs that form a continuous membrane organelle in the cytoplasm of eukaryotic cells and play an essential function in protein synthesis. This separates it from the other primary form of endoplasmic reticulum, the smooth endoplasmic reticulum, which lacks ribosomes and is involved in synthesizing and storing lipids both superficially and functionally.
The ninth and tenth part is nucleus and nucleolus. We used boiled eggs to serve as our nucleus and nucleolus. The nucleus directs and regulates the cell's functions, such as development and metabolism, and houses the genes, which hold the cell's genetic information. Small entities found within the nucleus are known as nucleoli. While the nucleolus is a membrane-free organelle within the nucleus that produces ribosomes, which are the cell's protein-producing structures, the nucleolus appears as a huge dark patch within the nucleus under a microscope. When chromosomes are brought together into nuclear organizing areas after a cell divides, a nucleolus is produced.
The 11th part is cell membrane. We use cabbage to serve as our cell membrane. The cell membrane, also known as the plasma membrane, is found in all cells and separates the cell's interior from the outside surroundings. The cell membrane is made up of semi-permeable lipid bilayer. The cell membrane controls the flow of materials into and out of the cell. The 12th part is cytoplasm. We use rice to serve as our cytoplasm. Cytoplasm of animal cells is a gel-like substance consisting mainly of water that fills the cells around the nucleus. It comprises proteins and chemicals that are crucial for the health of all cells. Salts, sugar, amino acid, carbohydrates, and nucleotides are all found in the cytoplasm of the animal cells.
The 13th is vacuole. We use relic to serve as our vacuole. A vacuole is an empty space within a cell lined by a membrane and filled with fluid. Vacuoles are crucial cytoplasmic organs or organelles in protozoa or single-celled eukaryotic organisms which perform processes such as storage, ingestion, digestion, excretion, and expulsion of excess water.
And that would be the end of our presentation. Thank you for watching.
The cell membrane controls what goes in and cell gets energy from the source of mitochondria, also known as the powerhouse. And the ribosomes of the cell with protein synthesis of the reticulum will carry substances breaking down materials, lysosomes. The nucleus controls the cell kinda like a baking things that DNA will tells the body to replicate. Inside the nuclear membrane full of nucleus make vital symbols and units from proteins secreting hormones like the Golgi bodies. Each unit of a cell is an organelle. The membrane keeps substances out inside cell building cytoplasm. Don't stop breaking down energy from mitochondria, protein, bacteria, ribosome, endoplasmic reticulum. Transfer substances like proteins and these controls they cell membrane, mitochondria, ribosomes, cytoplasm. protein synthesis, endoplasmic reticulum will carry substances making gamma to rebosomes. The nucleus controls the cell, kinda like a fake contains the DNA when sulfide to replicate. Besides the nuclear membrane full of the nucleus make ribosomes and units from proteins secreting hormones like the Golgi bodies. Each unit of a cell is an organelle. The membrane keeps substances out inside cell film inside cytoplasm. Don't break things down, energy from mitochondria. Protein factories, ribosomes, endoplasmic reticulum. Transfer substances like proteins, nucleus controls them. Mitochondria, ribosomes, nucleus, ARN, Golgi body, the nucleus, cell membrane, mitochondria, ribosomes, nucleus, ARN, Golgi body, the nucleus. Look at a plant cell, they have a few different organelles, chloroplasts, vacuoles, and a cell wall. Chloroplasts take the sun in, push the fluid through photosynthesis, large vacuoles of blue water and glaze. Cell wall is the outer, rigid cellulose as the protector, to provide structure to the cell. Cell membrane, mitochondria, ribosomes, cytoplasm, nucleus, ARN, Golgi body, the nucleus. Cell membrane, mitochondria, ribosomes, cytoplasm, nucleus, ARN, Golgi body, the nucleus. Also known as the powerhouse. Ribosomes have the cell with protein synthesis, endoplasmic reticulum will carry substances making gamma to ribosomes. The nucleus controls the cell, kinda like a brain contains the DNA and sulfide to replicate. Besides the nuclear membrane full of nucleus make ribosomes and units from proteins secreting hormones like the Golgi bodies. Each unit of a cell is an organelle. The membrane keeps substances out inside cell film inside cytoplasm. Don't break things down, energy from mitochondria. Protein factories, ribosomes, endoplasmic reticulum. Transfer substances like proteins, nucleus controls them. Cell membrane, mitochondria, ribosomes, cytoplasm, nucleus, ARN, Golgi body, the nucleus. Cell membrane, mitochondria, ribosomes, cytoplasm, nucleus, ARN, Golgi body, the nucleus. Look at a plant cell, they have a few different organelles.
A plasma membrane, or the cell membrane, provides a protection for a cell. It also separates the interior of the cell from the outside environment. And that membrane has several different functions. One is to transport nutrients into the cell, and also to transport toxic substances out of the cell. There are some facts about the plasma membrane. First, plasma membrane, it is made up of a double layer of phospholipids that separates a cell from the outside world. Second, it contains proteins that provides a number of critical functions. And the last fact is it contains carbohydrates that help to identify the cell and link the cell to others.
The Golgi apparatus, also called Golgi complex or Golgi body, is a membrane-bound organelle found in eukaryotic cells. Cells with clearly defined nuclei that is made up of a series of flattened stalk pouches called cisternae. It is located in the cytoplasm, next to the endoplasmic reticulum, and near the cell nucleus. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to targeted destinations. The Golgi apparatus was observed in 1897 by Italian cytologist Camillo Golgi.
Lysosomes was discovered by Belgian cytologist Christian de Dobe in the 1950s. According to him, lysosome is a cell organelle organate from membrane of Charles Golgi. Complex a lysosome subcellular organelle that is found in nearly all types of eukaryotic cells with an clearly defined nucleus and that is responsible for the digestion of macromolecule. All cells part in microorganism within lysosomes is surrounded by a membrane that maintains an acidic environment within the interior via a proton pump.
Cytoplasm is a thick solution that fills the cell and is enclosed by the cell membrane. It's mainly composed of water, salt, and proteins. All of the organelles in eukaryotic cells such as nucleus, endoplasmic reticulum, and mitochondria are located in the cytoplasm.
The smooth endoplasmic reticulum. It's a membranous organelle found in the most eukaryotic cells. The smooth endoplasmic reticulum functions in many metabolic processes. It synthesizes lipids, phospholipids, as in plasma membrane, and steroids. Cells that secrete these products, such as cells in testes, ovaries, skin, oil glands, have an excess smooth endoplasmic reticulum. Did you know who discovered the smooth endoplasmic reticulum? It was discovered independently by Porter. The name was given by Porter in 1953.
Ribosomes are small spherical organelles found in the rough endoplasmic reticulum that are responsible for the production of proteins. When proteins are manufactured incorrectly, they can end up in the endoplasmic reticulum they held, and the endoplasmic reticulum enlarges as a result of the proteins not getting out where they were supposed to go to.
Ribosomes are cell structure that makes proteins. Proteins is needed for many cell functions such as repairing damage or directing chemical processes. Ribosomes can be found floating within cytoplasm or attached to the endoplasmic reticulum. There are some interesting facts about ribosomes. The in ribosomes came from ribonucleic acid or RNA, which provides the instruction on making proteins. Ribosomes are different from most organelles in that they are not surrounded by a protective membrane.
The nucleus is the largest, most prominent organelle inside the cell. The functions of the nucleus are that it houses genetic material or DNA. It is the site of RNA production, and it helps regulate cell metabolism by generating various enzymes. The nucleus is the largest organelle in the human body, occupying around 25% of the cell volume. The structure of the nucleus can be divided into four main parts: the nuclear envelope, the chromatin, the nucleoplasm, and the nucleolus.
The nucleolus is the biggest structure in your eukaryotic cell. Nuclei is the most recognized for being the birthplace of ribosomes. Nucleoli are also involved in the creation of signal recognition particles in the cell. Stress response.
A mitochondria is a double membrane band organelle found in the most eukaryotic organism. Mitochondria generate most of the cell supply of adenosine triphosphate used as the source of chemical energy. They were first discovered by Albert von Kolliker in 1880 in the voluntary muscles of insects.
Centrioles. Centrioles are paired barrel-shaped organelles located in cytoplasm of an animal cell near the nucleus envelope. Centrioles are very important part of centrosomes and play a role in organizing microtubules, serve as cell skeletal system. They help determine the location of the nucleus and other organelles within the cell. Trivia about it is, centriole is derived from spirochetes, which is a bacteria within microtubules. The centrioles microtubules act as the part of a cytoskeleton of it.
The materials that makes up a chromosome, which is made up of DNA and protein, is called chromatin. Histones are the most important proteins in chromatin. They serve as DNA packaging components. The importance of chromatin stems from the fact that it's a pretty good packing method for getting all the DNA inside the cell.
Microtubules extend throughout the cell, providing it with proper shape and keeping the organelles in place. They are the largest structures into the cytoskeleton and are about 24 millimeters thick. They facilitate cell movement, cell division, and transportation of materials within the cells. They are also involved in the division of chromosomes during the process of mitosis and in locomotion. Microtubules are arranged in the form of microtubule organizing centers. They are structures found in eukaryotic cells. During the interphase, most of the animal cells consist of microtubule organizing centers. Several proteins are bound to microtubules, namely dynein and kinesin. The important functions of microtubules are cell movement, cell division, and cell transportation.
Here's what a cell contains. Outer layer is the cell membrane.